Hi everybody, welcome to another Chats with Chaudhry. I hope you're safe and well. Today I'm delighted to be joined by Bonnie Alden, who is the Principal Scientist at the Chemistry Technology Centre for Consumable Separations at Waters Corporation. And we are going to be talking about mixed mode chromatography for small molecule therapeutics. So hi Bonnie, how are you?
Pretty good this morning. How are you, Rizwan?
I'm very well, lovely to see you, Bonnie. I understand you're calling in from Boston. Hopefully the weather is a bit warmer now.
It's cooler this morning, but by the end of the week, it's supposed to be 85. So I'm already planning a weekend.
It's a difficult, isn't it? You never know what clothes to wear on each day, do you, with this changeable
I do
weather?
not.
Right, so before we start talking about the wonders of mixed mode chromatography, specifically around small molecule therapeutics, could you give people a quick overview of your background?
Sure. I have worked at Waters, for over 35 years, and I've worked on a number of different projects. It's always been chromatography related. I came in originally for sample prep development, and I have migrated over to actually synthetic materials development for chromatographic columns. So I'm the person that's probably before the customer. I'm not.
Fantastic. So you make sure everything works properly.
I make everything work properly, as well as we design it for its fit purpose, make sure it's fit for purpose for the customer base. So if we're looking at a column that we want to make sure can retain hydrophobic analytes, we'll make sure that happens with a lot of test cases.
Brilliant. So you are perfectly qualified then to talk about the subject. So let me ask you a very basic question to start. Can you give a brief explanation of mixed mode chromatography and how it's different from reverse phase?
Sure. As I mentioned that I have run all the types of modes of chromatography. So we have reverse phase, which is predominant mechanism for that retention is hydrophobic interaction with the analyte. I've also done ion exchange, and that is mostly a kind of a lock and key mechanism for the analyte. So you're basing it on a charge state.
Right.
And for mixed mode, actually you have two mechanisms of interaction, and they can be reverse phase as well as anion exchange, as well as maybe even a third interaction.
Right.
So most modes of chromatography are just based on one type of interaction, but mixed mode actually, like the name implies, there's multiple modes of interaction you can kind of leverage to get your best separation.
And is it more preferable in certain types of applications to others? And if so, what types of applications would it be more suitable for?
So one of the things with, say, reverse phase chromatography, which is a go-to, and you can actually do a lot with reverse phase chromatography, is that at some point you're going to hit some limitations. And those limitations could be how low you can go to retain an analyte in organic concentration of your mobile phase, or if you've got a mixture of different analytes in your sample that have different types of functional groups, like hydrophobic groups, or ionized acids, or even basic analytes all within one sample matrix, you're going to need some other type of interactions as well. Right. To separate them and retain them. So I'm looking at samples that say maybe have mixed functional groups available to them.
Fantastic. All right. Now, I understand that you set out to explore mixed mode separations for, and I may pronounce these incorrectly, so forgive me if I do, but for Umifenovir,
Umifenovir.
All right, and Lopinavir, and Ritonavir. I can't pronounce these. And Remdesevir. I should be able to pronounce these. Right, so you were looking at it. So can you give me a background why you decided to do that, though, in the first place?
Well, with the COVID-19 pandemic, one of the major focuses for them was to start looking at evaluating maybe small molecule therapeutics that could alleviate the disease or stop the disease, and repurposing small molecule drugs that have already been introduced for other types of diseases or conditions like SARS-CoV-1, the MERS virus,
right.
influenza. And you start looking at that kind of spectrum of molecules, and you're going to have all different types of functional groups on there. And you might be looking at them in different types of situations and different matrices or different, sample makeups.
Right. Okay. So your mixed mode approach, what were the advantages of the mixed mode approach for looking at the separation, for example, in Ritonavir or Lopinavir?
Okay, for Ritonavir and Lopinavir. So
Glad you said it properly.
that's okay. I might not be saying it. I don't ever hear them spoken out loud. I just try to keep that, I guess, stress on the last part, that actually the Navir is actually how you identify an antiviral type analyte.
Well, I'm learning new things all the time, so there you go. I'll remember that now going forward.
They're both antivirals, and they're both protease inhibitors. And
Right.
one of the things I actually noticed about them is they both actually have large log p-values. So,
Right.
and that's
Okay.
the octanol water coefficient. So they're in excess of maybe 4.5. So they're pretty hydrophobic. So I thought they may work fine on our reverse phase materials as well as the mixed mode. So I did a full column screen, and the mixed mode material did give me a little bit more retention. So I'd like to stay in that arena, if as much retention as I can get. That's kind of reasonable, nice resolution. And what I wanted to do is, if I wanted to look at maybe impurities in those drugs, that I'd have like room to work. So I kept that mixed mode.
Right. Okay. And what about for Umifenovir?
Umifenovir, you can say
Yeah.
its trade name Arbital.
Arbital.
And
Right. Okay.
you know, that was just a shot. I mean, that one came out early in the pandemic, and what I would do is I would read the literature and what was getting the most clinical trials or the most play in the press. It's like, let me take a look at that analyte right away. And I personally didn't wondered about that analyte because it is a base. It does have a basic site on it, and the column that I'm using is an anion exchanger as well as a reverse phase material. So I did not know if that would look really well on a mixed mode, but one of the benefits of this mixed mode material is at low pH, the mixed mode moiety on it, it does have a positive charge. So then the Umifenovir or the Arbital peak also has a positive charge at low pH. And it kind of, I want to say, ionically repels that peak. So it's a nice sharp peak. It's tall, it's thin. So it's a little side benefit. It wasn't what the material was particularly developed for, but it is something that actually is kind of like a really pleasant benefit on the end if you got a really sharp peak for some basic analytes. And that's what I
Right.
was looking at. So
Fine. And what about Remdesivir?
Remdesivir came out very early. It's a molecule, and it's a polymerase inhibitor actually. And what that does is just stops the viral replication. So there's a lot of ways antivirals work, and that's one of the ways. And the Remdesivir molecule itself is a prodrug. So one of the things I knew is that the prodrug possibly could be analyzed by reverse phase chromatography. So for my mixed mode, I do have that reverse phase functionality on it, so I could use that. So that was my first I want to say hurdle to get over, is to make sure I could look at Remdesivir, because I knew the biggest hurdle was going to be something that we did develop this column for, and that was the triphosphate. So your body will take that prodrug and metabolize it into the triphosphate, the active form. And those are the drugs that, those are the functional groups that don't get retained on a traditional conventional like alkyl based C18 column. You need to have that ion exchange moiety, that mixed mode feel. So we have tertiary alkylamines on there as well, and that's what's going to help me retain that triphosphate metabolite.
Right. Okay. Fine. So what would you say are some of the learnings you've got out of this, in a general basis?
A general basis. Well, one of the things is that these are drugs that have all been introduced before. So I was getting a nice spread of different analytes to look at. I mean, it was for the COVID-19 research, which was great. And the amount of research that was coming out and what people were looking at and why was very interesting to me. But some of the basics I could come from this work is that we talked a little bit about Umifenovir, and that's a basic drug. So if your mix has a basic drug in it or a basic functional group, you might be able to get some nice sharp narrow peaks, and that increases the sensitivity, so it'll be a taller peak, so you can actually look at lower levels.
Right.
And for say Remdesivir and Remdesivir triphosphate, I know how to actually tune the material now and get to maybe a good spot for analysis of both prodrug as well as the triphosphate metabolite, because you will find researchers that want to look at both in a patient. You want to see how that drug is being metabolized over time.
Right.
So we've learned how to tune the mixed mode to actually give some nice retention and some decent peak shape. The third one, I just found it an interesting, I want to say an interesting puzzle when you look at it, because there's so many different functional groups, and I was wondering, how can I bring mixed mode into like more of a mainstream use? It's not maybe well understood yet, but it has been getting more popular over the last several years, especially when you start getting very difficult pharmaceutical applications.
Sure.
Like recently I've looked at something called a polypill, and you probably don't take many medications yourself, nor do I. So I was amazed at a lot of drug companies now are formulating pills that have everything in them. So if you're taking a heart medication, 90% of patients will also take these other 4 medications.
Right.
And one of them might be basic or acidic or hydrophobic, and they put them all in one pill. And a lot of times you can do an analysis on like the production of that pill in one sweep.
Right. Okay. So if people want to get more information, where can they get more information on the work that you've been doing?
As it happens, I'm actually doing a webinar coming up this Thursday. I've done one, and I've got a lot more explanation of mixed mode chromatography in it and what it does. We have journal articles as well. And there are some application notes.
Fantastic. Well, Gon, you want to say sorry?
What was that last bit?
No, I thought you were going to add something else.
Oh, no, sorry.
No, well, what I was going to say is that if viewers, if you want to know more, all the links that Bonnie just mentioned will be above the video. Hence the reason Bonnie's not mentioned them all in detail. I will put the links above the video so that you can click straight through to the webinar and also to the application notes and to the website as well. So Bonnie, anything else you'd like to add about mixed modes before we finish?
I really think, it offers some great selectivity. I mean, the mixed mode that I'm using is the Atlantis Premier column, and that's a reversed phase anion exchanger. But there are other mixed mode materials out there, but mine is actually great for polar acids, so really hydrophilic analytes, and hydrophobic analytes. So we've done a new particle on it and a nice bonding, and they're actually as reproducible as your traditional C18s at this point.
Brilliant. Okay, so as Bonnie said, if you'd like to know more, and she obviously knows what she's talking about, so I'd recommend you check out the webinar. Make sure you click on the link above the video. There'll also be application notes, as I said, links above the video, and there's also the Waters website. So Bonnie, all that's left for me to say is thank you very much for taking time out for talking to me today. It was lovely to meet you.
Thank you.
And to viewers, I hope you found that useful. If you'd like to know more about mixed modes, as I say, check out the links above. And until next time, as always, stay well and stay safe. Bye-bye.